Hello, Annabelle.
Hello.
Hello, Annabelle. Welcome to Filo. Hi, I'm Rima, a bio tutor on Filo.
Hello.
May I know what is your question?
How could you change your translation step to more accurate represent protein synthesis?
Okay, how could you change your translation steps to more accurately represent protein synthesis? So, Annabelle, do you know what is protein synthesis? Do you know?
No, I don't know.
Okay, protein synthesis basically it is a part of central dogma of molecular biology. Okay,
I'm
do you know
sorry, I
what is
couldn't.
central dogma? Do you know what is central dogma?
No,
Okay, I will tell you. Do you know something about nucleic acid, DNA, and RNA?
no,
Hello, Annabelle, do you know?
no.
Okay, so basically, nucleic acid acids present in the nucleus of a cell. Okay, are you getting?
I can't really hear.
Look, DNA is the nucleic acid that is present in the nucleus of a cell. Okay,
Okay.
so this DNA is converted into RNA. Okay, and then this RNA is converted into protein. So this is called as the central dogma of molecular biology. Okay,
Okay.
okay. So there are two processes involved in this.
Okay.
Yes. So the process by which DNA is converted into RNA is called as transcription. Okay, and the process by which RNA is converted into protein is called as translation. So after this, we will learn about the steps of protein synthesis. Okay. So how could you change your translation steps to more accurately represent protein synthesis and describe why protein synthesis needs to occur? Okay, so the protein synthesis needs to occur to represent the gene expression. Okay, Annabelle?
Yes.
Okay, so here we will start with translation. Okay,
Okay.
so as you know, the process of formation of polypeptide chain from RNA is called as translation. Process of forming. Okay, so that's I show you here DNA is converted into mRNA, that is messenger RNA. Okay,
Okay.
so the process of forming polypeptide from mRNA is called as translation. Okay, did you understand, Annabelle, what is translation?
Mm, I'm still a little.
Did you did you understand? Is there any confusion? Hello.
Yes, I don't know what's mRNA.
Okay, so mRNA is a type of RNA. As you know that DNA is the full form of DNA is deoxyribonucleic acid and RNA is ribonucleic acid. So both these are type of nucleic acid that are present in the nucleus of a cell. And there are three types of RNA. First one is messenger RNA, second one is tRNA, and the third one is rRNA. So in the process of transcription, this DNA is converted into mRNA, that is messenger RNA. Okay. Is it clear now?
Yes.
Okay. So the translation is the process of forming polypeptide, means protein from mRNA. Okay. And if we talk about the steps of the transcription, mainly there are four steps involved. Okay. So here the first one is tRNA charging. First step is called tRNA charging. Second step is called initiation. Third step is called as elongation. And the final step is termination. Okay.
Okay.
Is it clear?
And
Is
yes.
it clear?
Yes.
Okay, so let's start with the first one, that is tRNA charging. I will send you the notes at the end of the session, okay?
Okay.
From there you can write. So in the process of tRNA charging, it is the process of attaching an amino acid to its respective tRNA. Okay, so what step occur in this process? Attachment of amino acid to tRNA. Okay,
Okay.
so this is also called as the amino acid activation. Okay, or amino acid acylation and tRNA charging. Is it clear?
Yes.
Okay. So Annabelle, the second step in this process is initiation. So what occurs in initiation here? The charged tRNA attached to the start codon. Do you know what is the start codon in the translation? Do you know, Annabelle, what is the start codon or the initiation codon?
I don't.
That is AUG. Okay. So here the charged tRNA. Here the charged tRNA attached to the start codon. So the start codon is AUG. Okay. Do you know what are codons? Do you know, Annabelle, what is codon?
No.
So basically, codon are the nitrogenous bases sequence. Here A, U, and G are the nitrogen. Okay, so here is the nitrogenous bases sequence. Okay, A for adenine, U for uracil, and G for guanine. Okay, okay, Annabelle, are you listening?
Yes.
Okay, so after attachment of the start codon, the subunit of ribosome bind to the mRNA. Okay, so
Okay.
what we will write here, ribosomal subunit bind to mRNA. Bind to mRNA. Okay, and finally the last ribosomal subunit bind to the initiation complex. So after initiation, here the third step is elongation. Okay,
Okay.
so according to this process, what happens? The codons that are found in the mRNA, so the polypeptide chain keeps growing. Okay, so here the growth of polypeptide chain occurs. Okay,
Okay.
here the growth of polypeptide chain occur, and here the each amino acid has a polypeptide bond that is attaching to the growing chain. Okay,
Okay.
so here the next final step is termination. So what happens during the process of termination when the ribosome attached to the stop codon? Okay, so that process of this translation terminate at the stop codon. So here the ribosome will bind to stop codon. Can you name the three stop codon, Annabelle?
No. A.
So the three stop codon are here UAAA, UAG, and UGA. Okay.
Okay.
Okay. So the process of translation is finished when these codons lack tRNA. So when this process happens, the translation stops and the newly produced polypeptide chain is released. So I'm sending you a diagram. Okay.
Okay.
So you can make the diagram only. Okay. If you don't want to write so much content, then you can simply make the diagram. All right.
Um. Okay. Thank you.
Just second, I am sending you the picture and I will explain you the picture also. Okay.
Okay.
Did you get the picture, Annabelle?
No, I. Uh. Now I did.
Okay. So I will explain you this picture now. Okay.
Okay.
So here what happens in this diagram? First of all, you have to make the diagram and you can write that content. Okay. So you will fetch good marks. So here, first of all, the tRNA with first amino acid. Okay, and here the anticodon is giving. Here, large ribosomal subunit is given in the brown color, and the small ribosomal subunit is given in the blue color, right?
Yes.
And at the 5' end, the initiating codon, start codon is present, and the 3' end, the UAG stop codon is present. So what happened during the process of elongation? You can initiation. Here the translational complex forms and the tRNA brings first amino acid in the polypeptide chain to bind to the start codon.
Okay.
And in the second step, elongation, here the tRNA brings first amino acid one by one to the polypeptide chain. So here the amino acid will be added to the polypeptide chain. And then finally, during the termination, here the release factor recognizes the stop codon and the translational complex dissociate means it's separate and it completed the complete polypeptide chain is released. Okay, so like this process is recycled again and again. Did you understand?
Yes.
Okay, Annabelle, do you have any other question?
No.
Okay, did you like?
Yes.
Okay, please rate the session if you like, and if you have any other question, then you can reconnect. Okay.
Okay. Thank you. Bye.
Okay, welcome. Have a nice day. Nice to talk to you. Bye.